Now, what are the constituents of epidermis? Epidermis actually is made up of, what does epidermis consist of? Like xylem, we said xylem vessel, xylem fibers, xylem parenchyma, tracheids, all that. So, in epidermis also, in epidermis the components can be recognized and they are of three types. First one is called as epidermal cell complex. Second one is called as stomatal complex, and third one is called as trichome complex. When we write an answer or when we are studying about epidermal tissue system, what we dealt earlier in this class is just an introduction to epidermis, where it is originating, what is the function of epidermis, what is about cuticle, what is about wax, what are the functions of cuticle, cutin, cuticularization, wax, all those are just comes under introduction. When we are talking about epidermal tissue system, we are supposed compulsory talk about three types of components of epidermis. When you are studying in detail about these three, we will complete your section or chapter of epidermal tissue system. In that the first one is epidermal cell complex, and the second one is stomatal complex, and third one is trichome complex. In epidermal tissue system, the epidermal cell complex, we will be studying about different types of cells. Those are called as epidermal cells, buliform cells, or costal cells. Now, when it comes to stomatal complex, in detail we will be studying about the structure of stomata, stomatal pore, guard cells, subsidiary cells, on the basis of position, how many types of stomata are there, on the basis of origin, and on the basis of arrangement of the guard cells and on the arrangement of the subsidiary cells also, how many types of stomata are there, that we will study in stomatal complex. In trichomes, what is trichome, how many types, unicellular, multicellular, uniseriate, biseriate, multiseriate, different types of trichome complex. What actually the trichome is helpful, this all will make a completion of your epidermal tissue system. The points for your entrance will come here is like costal cells, buliform cells, epidermal cells. In stomata you will get points on types of stomata, different types of stomata, and trichomes, which type of trichome is present in which type of plant. On the basis of example basis you will get the questions from this type. So, this will be giving you bit of 2 to 3 question bits from the MCET point of view. The first one we will be dealing here will be epidermal cell complex. Constituents of epidermis. In the constituents of epidermis, we will be studying about 3 different type of components: epidermal cell complex, stomatal complex, and trichome complex. So, now here we will be dealing with epidermal cell complex. In epidermal cell complex, we will talk about 90% of the epidermal cell complex or epidermal cell complex is occupied by the epidermis in total. Now, in case of epidermal cell complex, it consists of 3 types of different cells. Those are called as epidermal cells, second costal cells, and third one is buliform or special epidermal cells. So, in epidermal cell complex, we will talk with reference to epidermal cells, costal cells, and special epidermal cells. So, first we will be dealing about epidermal cells. Most of epidermal cells, most of the plant body or most of the epidermis is covered or made up of type of cells. Those are called as epidermal cells. These are living. These are compactly arranged. These are compactly arranged without intercellular spaces. Now, this central part of these cells is having a nucleus surrounded by the cytoplasm. Chlorophyll or chloroplast are absent. Chloroplast are absent. Hydrophytes and zeophytes. You will see the presence of chlorophyll. Normally we don't see the presence of chloroplast and chlorophyll in the epidermal cells, but in rare cases like hydrophytes in geophytic plants you will see the presence of the chlorophyll. And in a transverse section in TS, the epidermal cells are present in the form of long cells. That when we see the transverse section of epidermal cells, you will see all the epidermal cells are occurring in the form of long cells. And in case of surface view, in this, the cells are isodiametric in nature. The epidermal cells, if you see, they are isodiametric, means they are having almost same diameter and are stretched longitudinally. Either they are isodiametric or stretched longitudinally. So in surface view, these epidermal cells are either you will see them having isodiametric in nature, or you may see all are stretched and arranged longitudinally. Depending upon the species, the outer wall of the epidermal cells either may be thin or thick. In the leaves of conifers, the cells, in the leaves of conifers, the cell wall of the epidermis is thick. Now the cell walls are either straight. The cell walls may be either straight or curved or wavy or sinuous. What is sinuous means? Either the cell wall may be U-shaped or V-shaped. Right? As we said earlier, depending upon the different types of cells, the cell wall may be of the epidermal cells, cell wall may be thin or thick. In cases of, for example, if we take an example of conifer leaf, conifers will show the arrangement of the cell wall is thick. Now, what is the structure of cell wall? Like normally what type of cell walls you will see in different plants? Either you will see the arrangement of cell wall in a straight form, curved, wavy or sinuous. Sinuous means U-shaped or V-shaped. Now, when we see the curved nature, normally we will see the curvature of the leaf is more in the lower epidermal layers. So, there is upper epidermis and lower epidermis. When we said that the cell wall may be curved, so the curvature of the leaves may be seen more in the lower epidermis when compared to the upper epidermis. In certain species, you will see papillae-like structures. In certain species, you will see papillae-like structures are also formed in the epidermal cells. For example, where do you see these papillae-like structures? You will see in the plant called viola. This might come in your entrance. Dash, in this plant, we see the curvature of the papillae-like structures in the epidermal cells. That is viola, and one more example is Calicarpa. These two are important. Either in Calicarpa or viola, you will see papillae-like structures present in the epidermal cells. Okay, this is about your first cell, epidermal cells. Okay, next when we are coming across. Okay, next one more type of cell we have is costal cells. Another type of cell is B1 is costal cells. What are costal cells? The epidermal cells which are present on the veins are called as costal cells. The cells which are present on the veins of a leaf are called as costal cells. Now, these may be occurring in many rows, or they may be arranged laterally or these may be arranged in rows, or they may be arranged laterally or parallel to the long axis of the cells. Now, costal cells are stretched longitudinally. Costal cells are stretched longitudinally and they differ from the epidermal cells. Cells on the midrib, we said that the cells which are present on the veins are called as costal cells. So, if this type of epidermal cells called costal cells, if they are present on the midrib, so they will be in a manner of 20 to 40 cells. It will be in the form of 20 to 40 rows of costal cells. You can see if it is in the case of midrib. If it is in the case of midrib. If this type of costal cells are occurring in the lateral veins, you will see occurrence of few rows of costal cells. Rows of costal cells will be a bit less. Either it may be between 15 to 20. Okay, so in midrib you will see the more number of rows of coastal cells are present. Okay, for example, for this is there is an example called as Nothosierva brachiata. In this, the coastal cells are present only on the midribs. You will see the presence of coastal cells only in the midrib. Okay, this is about coastal cells. Coastal cells are the cells which are present on the veins of the veins are called as coastal cells. They occur in the form of rows. They are arranged laterally or parallelly to the longitudinal axis, and these are much better than the these are stretched longitudinally. They differ from the epidermal cells. On midrib, you will see occurrence of coastal cells in 20 to 40 rows. In case of lateral veins, it will be in very few rows compared to the midrib. So now this coastal cells, for example, you will see in the plant Nothosierva brachiata, you will see the occurrence of coastal cells in maximum number in midrib. Okay, next one more type of epidermal cell complex tissue is special epidermal cells. Okay, in epidermis, in some epidermal cells, here and there certain materials are deposited or certain materials are included, and these differ from other epidermal cells, and these are called as special epidermal cells, and the other name for this you can call it as idioblast cells. And these are differing from other normal epidermal cells, and these cells are also known as idioblast cells. Examples can be taken as tannin cells, myrosin cells, mucilage cells. All these come under special epidermal cells: idioblast cells, mucilage cells, tannin cells, myrosin cells. Beside this, in many angiosperms, the crystals are beside this. These are special epidermal cells, of which idioblast. And in case of angiosperms, we will see the presence of some crystal-like structures of very much crystal. In angiosperm, we will see some crystals-like structures which are occurring in different forms. Some are in needle-like form, needle-like crystals. So these crystals which are needle-like, either they occur in singular form or in the groups. So these needle-like structures are called as, needle-like crystals are called as raphides. This is important. In angiosperm, special epidermal cells or idioblasts are present, which are like in the form of crystals, and these crystals will have different shapes. And in this, in some angiosperms, they are of needle-like structures. So these are needle-like special epidermal cells which are present are known as raphides. Now, what are these raphides made up of? These raphides are made up of calcium oxalate. In addition, one more special type of epidermal cell complex or special epidermal tissue cell is present. Those are called as cystoliths. Now, what are cystoliths in epidermal cell, special epidermal cells under epidermal cell complex? One more is there, that is called as cystoliths. In cystolith, the example here we can take it of leaves of Ficus elastica. For example, in the leaves of Ficus elastica, you will see the presence of special epidermal cells which are called as cystolith, which hang from the stalk, and appear as if there are a bunch of grapes. Special epidermal cells are present, and these are appearing as if it is a bunch of grape. These are normally seen in the leaves of Ficus elastica. And what are these cystoliths made up of? These are made up of crystals of calcium carbonate. Okay, and one more is there, that is called as spheroraphides. Raphides and spheroraphides. Now, another, we have studied about the raphides, cystoliths. Now we will study about spheroraphides. Now what is spheroraphides? These are also a type of crystal like structures which are appearing in the form of stellate structures. Spheroraphides are also called as druses. Another type of special epidermal cells which is present is called as spheroraphides. The alternative name for this is called as druses. Now these druses are nothing but these are also crystal like structures, but these are in the stellate form and these are occurring in many angles. These are stellate with many angles. These are stellate with many angles. So, how many special epidermal cells we have studied now? First one is epidermal cells which we have studied is, first is idioblast. Example for idioblast was myrosin cell, tannin cells, mucilage cells. And another one is one more in angiosperm we have studied some needle like crystal structures which are in the form of needle. Those are called as raphides, which are made up of calcium oxalate material. Now another one more example of special epidermal cell we have is cystoliths. In cystolith, like these are those structures which form from the stalk and they appear in the form of a bunch of grapes, and these are made up of crystals of calcium carbonate, and normally these are seen in leaves of Ficus elastica. Now one more type of special epidermal cell we are supposed to study was spheroraphides. The alternative name for spheroraphide was druses, and these are nothing but these are also crystals which are in the form of stellate with many angles in them. Example, these are also made up of calcium oxalate. Examples for spheroraphides are you may get question on what are druses. Examples for druses. You may get question on amaranthus. Special type of epidermal cells present in the amaranthus. What are druses? This type of questions you may get. And these are in some cells. For example, in there are some more specialized epidermal cells in which there is a deposition of inulin. In some epidermal cells, inulin also is occurring. These are deposited. The occurrence or presence of inulin is occurring in the form of fan shaped specialized epidermal cells are present where the deposition of inulin is occurring. These are also seen in an example of dahlia. So this is important. Examples are important major. Cystoliths ke example, leaves of Ficus elastica. And another one, spheroraphides ke examples, Carica amaranthus. And there are some more specialized epidermal cells where the deposition or storage of inulin occurs, and the shape of that epidermal cell is in the form of fan, and those are examples of dahlia. So these special why we are studying these epidermal cells, special epidermal cells, because these all special epidermal cells are basically having a major impact or more significant in the classification of the plant. Okay, so now here these are about special epidermal cells. Now one more is there, epidermal cells in grasses. We talked now about different types of epidermal cell complexes. Now we will talk about epidermal cells in grasses. So what are the different shapes, what type of epidermal cells are seen in the grasses? Like for example, in a family like Poaceae, the epidermal cells are differentiated into two types. Those are long cells and short cells. Okay, these are occurring, which are occurring in parallel rows. There is one long row or long cells row, then is short cells. So both are occurring in parallel rows. And now again these short cells are again divided into two types. Those are called as silica cells and Koch cells. Right? When we talk in terms of grass, the epidermal cells, we can take an example of a family called Poaceae. In a Poaceae, the epidermal cells are differentiated into two types. One is long cell, another one is short cell, and which are occurring parallel rows, which are occurring parallel to each other in parallel rows. And again, when we go further, these short cells are again further divided into silica cells and Koch cells. Now when we talk about silica cell and Koch cell, how they occur? They occur they are lying side by side and one above the other. The silica cell No, we said short cells are again differentiated into silica cells and Koch cells. So, for example, if it is the structure showing some Koch cells, presence of Koch cells and silica cells. So, we see here these silica cells and Koch cells are occurring one above the other side by side. Now, when we talk about the silica cells, these cells consist of structures called as silica like structures are present here, and these structures which are present are either in surface view. When we see the structures of silica which are present in the silica cells, you will see that these may be either round, these may be circular or elliptical in structure, or either it may be dumbbell in structure, or it may be in the form of saddle like structure. So, the shape of the silica like structures which are present in the silica cells in surface view, they may be either circular, elliptical, dumbbell shaped or saddle like structures. Now, what is the use? Because of the presence of silica cells, the structure or the roughness of the leaf will be showing the rough texture. The roughness of the leaf is because of the presence of the surface of the leaf is becoming rough because of the presence of certain structures called as silica cells. Now, when it comes to Koch cells, what are Koch cells? Koch cell consists of suberized cell walls. It consists of suberized cell walls. It means these are consisting suberin in them. These are solid organic materials which are present. These Koch cells and silica cells are very few in number, and these are growing in those cells which are having those leaf like structures which are having rapid growth ability. Then we can see the presence of silica and Koch cells, and these are stomata which are present in between the epidermal cells. Okay. Now, next one we are having is, this is about epidermal cells in grasses. Example is poaceae. In poaceae, it is divided into two types, long cells, short cells. We are talking in terms of short cells here. It is of two types, silica cells and coastal cells. Silica cells and coastal cells lie one above the other side by side. Silica cells will have stalk like structures, silica like structures which are present. When we see in surface view, these are circular, elliptical, dumbbell or saddle like structure. The leaf surface is rough owing to this type of structures. Now, when we come to Koch cells, it is consisting of superized cell walls, and these with solid organic material present in them. This silica occurs in the young cells. Silica cells occur in the young cells of the leaves and very few in number, and these silica cells and Koch cells are normally seen to appear in the leaves or in the cells which show rapid growth in them. Buliform cells. Okay. Now, another type of epidermal cells which are present, those are called as buliform cells. Now, why buliform cells? Where does this present are occurring? These are normally seen in monocots. All the epidermal cells which are specially seen in the monocots are in the those epidermal cells are not called as epidermal cells, we call them it as buliform cells. But except in the Graminaceae group, except in halobials, except in this, remaining all Graminaceae members will show the presence of buliform cells. Now, these are larger in size when comparing to the normal epidermal cells. These buliform cells are also called as motor cells. These buliform cells are bubble-like cells. Builliform cells are also alternatively called as motor cells. These are bubble-like cells. In transverse section, they are, if you are taking the TS of the builliform cells, they appear in the form of fan-shaped structures. In transverse section. Okay, in halobials there is no builliform cells. In monocots, the epidermal cells called as builliform cells, except in halobials of Gramineaceae family. Except halobials, all are having builliform cells. It means in halobials members there is no presence of builliform cells. Builliform cells are comparatively larger in size to the normal epidermal cells. The alternative name for builliform cells is motor cells. These are in the form of bubbles, so these are called as bubble-like cells. And if you see the transverse section of the builliform cells, you will see that these are in the form of fan-shaped structures. Now, the cell walls here are made up of, cell walls are thin and made up of cellulose and pectin. Now, cells are large, vacuolated, cells are large and filled with water. These are hyaline without chloroplast. And what is the use of these cells? These cells are helpful in the rolling and unrolling of the leaves. Now, we said that what is the use of these builliform cells in the leaves? These builliform cells helps in the rolling and unrolling of the leaves. Why this rolling and unrolling of the leaves occurs? Because this is because of the change in the turgor pressure of the leaves. We can see that as there is a change in the turgor pressure of the leaves, we can see that the rolling and unrolling of the leaves is occurring due to the change in the turgor pressure which is existing in the leaves. Now, when we say about builliform cells, normally we will see in the case of leaves. So here, builliform cells are mostly seen in the upper epidermal cells. And these are present in the form of parallel strips. Parallel strips alternating with the veins. Okay, now in majority of the monocot plants, you will see the presence of builliform cells in the upper epidermis cells, and in some plants you will see the presence of builliform cells in lower epidermis also. Okay, this is about builliform cells present in monocots but absent in halobials, also called as motor cells, also called as bubble-like cells because they are in the form of bubbles. In TS, we will see it in the form of fan-shaped structure. These cell walls are thin, made up of cellulose and pectin. Cells are large and vacuolated. These are hyaline in nature without chloroplast, and this majorly helpful in the rolling and unrolling of the leaves because of the changes in the turgor pressure of the leaves. Normally, where do you see the occurrence of the builliform cells? We will see this type of builliform cells majorly you will see in the occurrence of on the upper epidermal cells, and these are occurring in the form of parallel strips in between the alternating with the veins, and in some plants they may also occur in the lower epidermis also. So with this we end with your constituents of epidermal cells. Okay, in this first one was special epidermal cell complex. In epidermal cell complex we have studied about epidermal cells, builliform cells, we have studied about epidermal cells, costal cells, and again we have studied about special epidermal cells, cystoliths, raphides, idioblasts, spheroraphides. We have studied about inulin storing cells. Again we have studied about builliform cells. So these are coming under first section epidermal cell complex. Okay, next we will be having is second type of epidermal tissue component, that is stomatal complex.